Thanks for everyone for coming, especially the people who had to get up at the obnoxious hour of 6 a.m. Kind of feels like a World Cup match where we got to set the start time to satisfy people on several continents, and I'll try to make it so it's not like a 0-0 contest where you're bored and unsatisfied at the end of it. So as I said, our goal here is species tree inference under the multi-species coalescent. What we use is a Bayesian-weighted assembly method. So our basic steps is that we start out with the assumptions of the multi-species coalescent model and the Jukes-Cantor substitution model. And from that, we can find the probability of sequence data given speciation times and species tree topologies. If we then treat speciation times as nuisance parameters, we can integrate over them to get the probability of the data given only the topology. And then from there, we can find posterior probabilities of each 4 or taxon species tree, and then use assembly methods to infer larger trees. This is joint work with Laura Kabatko, also at Ohio State. This is recently published in the Bulletin of Mathematical Biology. And the raw code is also available on GitHub. It's currently only in the raw code form. It uses thread building blocks for parallelization. And so I've had some issues getting it to create executables that anybody can just get the executable and run with it. And so if you want to use the raw code or want to use the executable, just shoot me an email and I can work with you for getting executables to run on your computer if you're interested. All right, so quick intro into our problem. Our goal here is to infer a species tree. What I mean by that is, looking at this visual description of a tree, we're interested in things like if we break here, what are the number of species that fall on either the left side or the right side of some partition of the tree. Also interested in the particular labels down here. In other words, is the human and the chimp more closely related to each other than either is to the mouse or vice versa? And we're also interested in the speciation times. So, what is the distance here, which we call tau, the speciation time in this example between a human and a chimp? So those are the things that we're typically concerned with this inference problem. Because of the limitations of our method, we can only focus on the topology, and we have to treat the speciation time as a nuisance parameter. So we cannot infer that with our methodology. As a more formal definition, we say species tree is an acyclic graph consisting of a vertex set, an edge set, and a vector of node times, where the external nodes on the tree represent existing species, internal nodes represent speciation events, and the interior branches of the tree describe a spectrum of ancestral species. The complication with this picture is that for various reasons, such as during meiosis, you have crossover, you have incomplete lineage sorting, gene duplication and loss, horizontal gene transfer, various reasons for where particular parts of the genome may have different histories from other parts of the genome and from the species tree as a whole. And so the history of these individual parts of the genome are described by what we call gene trees, where once again, a gene tree is an acyclic graph consisting of a vertex set, edge set, and node times. However, with the gene tree, the eternal nodes represent coalescent events, where the coalescent event is the most recent common ancestor of a pair of gene lineages. And these node times, these coalescent events, are subject to the constraint that the divergence must occur prior to the divergence time of the species in question. And so here's a picture of a gene tree evolving within a species tree. So our coalescent time would be this node here. That's a coalescent event. And clearly this coalescent event has to occur further in the past than this speciation event between humans and chimps. And so we measure time going backwards as in typical. So typically the mathematical restriction is that this coalescent event, which is described by a t, the t has to be bigger than the tau, which is the mathematical label for the speciation event. And so not all possible gene trees can occur because of these constraints. And typically from our perspective, we treat these individual gene trees as again, a nuisance parameter that we can integrate over to get the probability of the data given solely the species tree and the speciation times. A last note on gene trees is that the name gene tree is something of a misnomer in the sense that, right, there does not necessarily need to be a one-to-one correspondence in either direction between the DNA sequences whose history is described by one of these trees and the biological sense of a gene being something that codes for a particular protein. So because of recombination, you can have multiple different lineages within the same gene, and conversely, in particular for mitochondria and the Y chromosome that are passed down without necessarily recombination, you could have multiple genes with a common gene tree. And so perhaps these should be better labeled as locus trees, but as far as I know, that name hasn't really caught on. And so the length of these recombination-free segments of the genome that are described by a single gene tree, as far as I know, that's still up for a matter of debate, but they can be fairly short, or in the case of, let's say, mitochondria, quite long. All right. Another sort of note, in phylogenetics, we have this concern of always identifiability in the sense that without knowing the mutation rate, it's hard to do inference on time. And so there are three major time units that we use. One is calendar time. The problem there is that without knowledge of the mutation rate, you can't really do inference on, the distance between species in calendar years. So another time unit that is used quite often is a mutation unit, which is the expected time to one mutation. So one mutation unit is the time you'd expect to take until there's one mutation at a particular site. The problem with that one is that leads to very, this is a very long time period. So one mutation unit is not very useful as a time unit. And so the third common time unit is coalescent units, where one time unit is the time to one coalescent event. And the way you scale between mutation and coalescent units is by this population parameter, which is called theta, where theta is related to the population size of the species in question, and where higher values of theta correspond to more mutation along the branches of the tree. And so coalescent units is a time unit that we use in our work, just because it's a more useful and practical unit of time measure. Okay, and so next I'm going to show the parameterization that we use in terms of species. And so on the left here, we have a 4-taxon symmetric species tree. On the right, we have a 4-taxon asymmetric. And again, the black, let's see if I can zoom in. Okay, we can zoom in here. So the black lines, the black outer lines describe the species tree. The green inner lines describe one example gene tree. These 3 taus here is the vector of speciation times. So you can see these correspond to the speciation times here, here, and here. And then the 3 T's, T1, T2, and T3 refer to the times of these 3 coalescent events. And then we use the labels A, B, C, and D lowercase to describe the different taxa of the speciation tree. Now we only need to consider the 4-taxon symmetric and the 4-taxon asymmetric tree because to find probabilities given any other labeling, so if we wanted to consider the probabilities given a labeling DCBA instead of ABCD, then we can just apply a permutation matrix to find those probabilities given the probabilities ABCD. And so we don't necessarily really need to concern ourselves with any other 4-taxon trees other than these two sort of model trees. And then in the 3-taxon case, it's similar, except you only have 3 taxa, so you only have 2 speciation events and 2 coalescent events to keep track of. And again, we have an example here of how we permute to find the data probabilities given given different labelings on the tree. Okay, so what is our data that we're using to do this inference? So the data is, blow this up here. So it's an example of sequence data. So this is example theoretical sequence data for 4 species, a human, a rabbit, a duck, and a cockroach. And we make the assumption that each of these aligned sites, so each column is an aligned site, each row is a taxa, and we make the assumption that each of these aligned sites are exchangeable. And if you make that assumption that they're exchangeable, then we don't need to concern ourselves with the data as a whole. We can reduce it down to where our data is just the number of sites that have particular site patterns. So the ones that are circled are AAAA. So all 4 species have adenosine at that site. And so we count up the number of times that occurs. And so the number of times we have AAAA, the number of times we have AAAC, and so on, all the way to the number of times we have TTTT. And that is for 4 taxa, 256 by 1 vector. And that's a sufficient statistic for our data probabilities. And then if we further assume the Jukes-Cantor substitution model, which says that all four nucleotides have the same limiting frequency and the same transition probability from, let's say, A to C as from G to T, then we don't need to concern ourselves with it, whether it's AAAA or CCCC. All we have to concern ourselves with is, are all 4 the same, or is the first 3 the same and the 4th one different, and so on. And I call that vector of site pattern frequencies D sub JC. And that is a 15 by 1 vector of site pattern frequencies. And given these assumptions, this 15 by 1 vector is a multinomial distribution given the total number of sites J. So J is our number of sites. And then this 15 by 1 vector of site pattern probabilities. And so our goal really is to infer this first off, this vector of site pattern probabilities. And once we have that, then we can do inference on the topology and things like that. Okay, so our approach to handling this multi-species coalescent problem is that if each site's history is a random draw from the space of all possible gene trees, then we can treat the gene tree history as a nuisance parameter to be integrated over. So our goal is to find that vector of site pattern probabilities given S, our topology, and tau, our speciation times. And for a given site, that is the sum over all possible gene trees consistent with species tree topology, which is given by that term script G sub S, integrating over the speciation times, and then a factor for the data probability given the gene tree and the species tree, and then the density of the gene tree given the species tree. And then if you further make an assumption of neutral selection, this term here, this mutation term, now only depends on the gene tree. And then these two terms here and here are independent of one another. And so this was calculated by Schiffman and Kabako in 2015 for 3 and 4 taxon trees. So they calculated this out, integrated over the gene trees, and found site pattern probabilities given speciation times and the topology. And so our new method, which we call LILY, which stands for likelihood-based assembly, is to now treat the speciation times as nuisance parameters as well. So we can place a prior on tau, and then we integrate over these taus to get delta given S. So delta given S is the integral over tau of delta given S tau F of tau d tau. So just standard law of total probability. And the name, the assembly part of the name comes from the fact that we can only really do this for up to 4 taxon trees. Site pattern probabilities for higher, larger taxon trees are theoretically obtainable, but have yet to be calculated. And there's just practical limitations in terms of computation time to integrate over all these possible, larger trees. So that's our approach. Quick comparison to other approaches. So our approach is to treat each site as if it were independent and integrate over those site histories. Other approaches, one is concatenation, which is to treat all the data as if it comes from a single gene tree, and then that gene tree that gets inferred is taken as your species tree. And this can be pretty fast and in some cases accurate because you're removing a lot. You're removing having to worry about all those gene tree densities from the equation. But the big problem with this is under certain conditions, it can be statistically inconsistent. So it can actually point you to the wrong tree, and increasing the amount of data you have doesn't help you. So that's really a problematic property to have. Another approach is to co-estimate both the gene and species tree together, which has the advantage you can generate, posteriors for all your parameters. If you're interested in different gene tree histories, well, it gives them to you. But the big limitation here is, it can be quite slow, especially if the number of taxa is large or the number of loci is large. The third method is what's called summary methods, which can infer the gene trees and then use the gene trees as inputs to infer the species tree, which is consistent, can be quick, which is good. But the real dependence here is the accuracy of the gene tree inputs. So if you can't delineate the gene trees properly and can't infer them because the size of the gene, the size of the recombination-free loci is small enough that you can't infer the individual gene trees accurately, then these summary methods can run into trouble. So that's sort of the layout of methods. So just intuitively, we would expect our method to sort of do better than these summary methods if we have a lot of independent loci and those independent loci are short, whereas the summary methods would tend to do better if the size of those recombination-free loci is larger. And so naturally, we simulated under a bunch of different conditions to see sort of like who wins under different conditions. All right. All right. So now a larger overview of our method. We have 2 different methods called LILY-T and LILY-Q. The T and the Q stand for triplets and quartets. So what LILY-T does is it infers posterior probabilities for each of the rooted triplets, of which there are 3 times n choose 3. And then it infers the rooted n taxon topology using an assembly method that takes those posterior probabilities as inputs. Lily Q is similar, except it infers posterior probabilities for each of the n choose 4 times 3 unrooted quartets. We can prove that the maximum a posteriori tree from Lily T approaches the true tree with probability 1 as you increase the number of sites under the condition of the strict molecular clock and the JC69 substitution model. And that's actually uniformly true for all triplets. And so you get the correct n-taxon tree. We can show that that should work through simulation for Lily Q. However, a formal proof is eluded us, unfortunately. So what the formal Lily T theoretical guarantee is that given independent sites, so given coalescent independent sites, for 3 taxa evolving under the multi-species coalescent with the JC69 model and the strict molecular clock, the probability that the inferred tree equals the actual tree goes to 1 as the number of sites goes to infinity. For any prior, that gives you a bifurcating tree. So as long as you say tau 1 is strictly greater than 0 and tau 2 is strictly greater than tau 1, that'll hold. All right. So the overview of the Lily T procedure. So I'm going to describe the Lily T procedure in some detail, and then just talk about the differences with Lily Q. So Lily T for each subset of triplets. So given n taxa, we take each n choose 3 triplet. We find the site pattern probabilities given both the topologies and the speciation times from Schiffman and Kabako. And like I said, then we can find permutations to find the probabilities given other topologies. Then given the law total probability, we integrate over the speciation times to find the site pattern probabilities given solely the topology. The prior that we assume for the speciation times is the Ewell model or the pure birth model, which assumes constant and equal speciation rates across all branches and times. And so given the Ewell model and the speciation rate beta, we say the first speciation time is an exponential 3 beta. The second, the difference between the first and the second speciation event is then exponential 2 beta. And that beta, we estimate from the data. And this gives the procedure for estimation of the speciation time. Essentially it just uses the Hamming distance between 2 species to estimate a speciation time, and then we just take the average of those to give us our rate. Don't want to get into this in too much detail because I want to move on. All right. So then the third step is given these site pattern probabilities, we can find the log likelihood of the topology given the data. And if we say each of the 3 possible 3 taxon topologies are equally probable in the prior, then the posterior is just the likelihood of one tree over the likelihood of all trees. So given by this equation here. And so then given the posteriors for each rooted triplet, we can find a rooted n-taxon tree using the assembly method of Sevilla et al. 2016. And so as far as an example, let's say we have 5 taxa, then we'll have 5 choose 3 or 10 different subsets. So the first subset is ABC, second ABD, ABE, and so on all the way down to CDE. And then we can infer hypothetically posterior probabilities for each of those 3 possibilities for each of the n choose 3 subsets. And so we have a set of 30 trees and 30 posterior probabilities. And then we can put it in the algorithm. And if you wanted to, you can show that all of these trees are consistent with the final, or compatible with the final n-taxon topology of A, then B, then C, then D and E. Okay, so that's the rough method by which this assembly procedure works. Lily Q is largely the same, except for the prior is slightly different in that we have 3 different speciation events instead of 4. And then for the symmetric topology, we have to assume that each of the first 2 speciation events occurs before the other with a 0.5 probability. And then the prior on the topologies is slightly different because the symmetric topologies have a higher prior probability if you assume the Yule model. And the last bit is that it turned out that the 15, so we get posteriors for each of the 15 rooted 4 taxon topologies, but finding the rooted 4 taxon topology turned out to be inconsistent. And so we sum the rooted 4 taxon topologies that correspond to particular 4 taxon unrooted topology, like you see here. So each of these 5 rooted 4 taxon topologies correspond to this unrooted topology. And so let's say if this had a posterior 0.2, this had a posterior 0.1, 0.05, 0.05, 0.05, we would assign a posterior probability to the corresponding unrooted topology of 0.45. And then we would do that for all 3 of the unrooted 4 taxon topologies and feed that in as an input to the final assembly procedure. And the assembly procedure that we use is from Abney et al. 2014. Okay, so that is the basis for the methodology. And so next I'll talk about some of the robustness checks that we ran, because we do make an awful lot of assumptions here. So we want to do a lot of robustness checks to show that this would be still work if our assumptions were not met, and then talk about the comparison to Astral, which is one of the summary methods, and where we could delineate where Astral did better and where we did better. So we relaxed a number of assumptions to see like how robust our model was to those assumptions being violated. The first off, instead of the JC69 substitution model, we simulated data under a GTR plus gamma plus I model, and that really didn't have any effect that we could notice. No real effect upon our accuracy. So we just ended up doing all our simulations using GTR. We tested under various values of that population size parameter theta. There really wasn't really any apparent effect for misspecifying. And theta tends to fall in an empirical range between around 0.01 and 0.001. So we just picked 0.003 as sort of the mid-range of the empirical range on the log scale, and just use that for all our calculations. Our model assumes independent sites, and so we simulated data using multi-locus data instead of independent sites. Having fewer loci did reduce accuracy. However, it didn't appear to reduce our accuracy that much more than any other method. So every method tends to do worse with fewer independent loci, because you just have less data. And so our model did worse, but really not substantially worse than others. Testing of that speciation rate, our knowledge of that speciation rate, really this is somewhat of an asymmetric effect in that if you overestimate the speciation rate, that can reduce the accuracy of your estimates. If you underestimate it, it's not as big of a deal. And that's just due to the fact that exponential curves are asymmetric. And so it turns out that our our estimation method for that beta is actually biased, and it's biased to underestimate the speciation rate. And so using that actually provides us with a little bit of a buffer against overestimating the rate, which is where our estimation method gets into trouble. We tested different speciation models, so different than the Yule model. We didn't really see to have any sensitivity to that. The last bit, we relaxed the molecular clock, and there the model is white sensitive. And in fact, it can be shown that given severe clock violations, our method will in fact be inconsistent. And so that's a known problem. And so we tried to highlight in our paper, hey, you really have to depend on the clock in order for this to work. And that's part of the future work section that I'll talk about at the end. Okay. And so for the large scale simulations, we simulated for different numbers of taxa, different values of the population parameter, different numbers of loci, different numbers of sites per gene, different speciation rates, performed 100 runs at each combination of settings, and compared both our new methods to SVD quartets and then to Astral, which is an example of one of these summary methods. Here are the different topologies that we used drawn to scale. And so we had some not very far apart in terms of time and some larger to time. And then this little red bar here indicates the scale. So either 0.1 coalescent unit all the way up to 2 coalescent units. So when we changed the size of that red bar, it sort of grew and shrunk the overall topologies accordingly. And so we measured how good our estimates were and how good the estimates of the various methods were at finding the exact tree. And our criteria was the mean RF distance, so Robinson fold, so the number of bipartitions that are different between the two trees, the symmetric difference in the bipartitions. And this is one example of the results for a mean or minimum branch length of 0.5 coalescent units, population parameter, population size parameter of 0.0008. The different colors represent the different methods. So gray is Lily Q, yellow Lily T, blue is Astral, and green is SVD quartets. And then this first number here is the number of loci. And the second number there is the number of sites per loci. And so we went all the way up to like 250,000 sites and as few as 5,000 sites. We did not include Astral for these two because Astral needs to infer a gene tree and it can't if you have independent sites. So we only compared against Astral for when we had multi-locus data. And so what we can see here is that the new methods outperform SVD quartets. Lily Q tends to outperform Lily T. The comparison against Astral is a little bit more mixed. In this display, Lily Q did a bit better than Astral, but at other settings, Astral did better than our method. And so as overall takeaways from our simulation results, all methods did better with more mutation because there's more signal. All methods did well when you had a lot of data. All methods did well with more independent loci. In general, SVD quartets tended to be outperformed by the other methods, and Lily Q tended to be better than Lily T. For long branch lengths, Lily Q generally outperformed Astral at 0.5 coalescent units. Astral outperformed for some settings, Lily Q for others. For shorter branch lengths, Lily Q did better when the length of those recombination-free loci was really short, or when the population parameter was low. Astral did better with longer gene lengths and larger values, so more mutation. Astral tended to do better. Okay, and then for uncertainty quantification, we performed bootstrapping. So we bootstrapped first the loci and then the sites within the loci. And what we have here is for Lily T and Lily Q, where it says node correct 1, that is the bootstrap value for a split that the method got correct, and 0 is a split that the method got incorrect. And you can see the bootstrap values are much higher when it got it right. And so that indicates that there's, bootstrap support is not a probability, but it says that when you have high bootstrap support, that's an indication that you got it right. And then we tried different ways of de-averaging this connection between bootstrap support and accuracy. And it turned out, so this is over here kind of an easier case, and this over here was a harder case in terms of, shorter branch lengths, less mutation. And then for the harder case, you get sort of a less of that splitting between the bootstrap support for the ones you got right and the ones you got wrong, where for the easier cases, it's clear that if the bootstrap support is low, that you probably got it, that you may not have gotten it correct. And then we did the same thing sort of in the other direction, where we said, hey, given the bootstrap support, how often did I infer the correct split? And again, over here, this is the easier case, and over here, this is the sort of harder case. And what we can show is that at no time did the bootstrap support be lower on average than your actual probability of getting the correct split or getting the split correctly. And in fact, if the tree was easier, your bootstrap support tended to really underestimate your actual accuracy. And so that's good, right? Because if we want to be conservative, we want a conservative measure of uncertainty, and this bootstrap support tends to provide that. We also ran these methods on some empirical data. So one is a 7-taxon data set of some snake species, 52 individuals from different snake species, both nuclear mitochondrial genes. And our results, we were able to do this in under a half hour, even with all these different, even with a lot of data, it ran fairly quickly. And our inference matched other methods. And here's another data set for 8-taxon yeast data set, 127,000 sites, over 100 genes. We completed the point estimate and bootstrapping in under a minute. And our results, again, sort of matched what was out there in the literature. So looks to be fairly accurate, and has a nice, certainly a speed advantage over like co-estimation methods, some of your Bayesian methods. Okay, so most of this work was done a while ago, just given publication timelines and things like that. So what have we done since then? The big one is generalizing the probability distributions to allow for different and time-varying rates for each branch of the tree. And so we've extended this for basically any rate function that's Riemann integrable. We can find now the probability given the speciation topology, the speciation times, and what I've been calling gamma, which is a vector of rates. So we've extended that. Still working. So we found the probability distribution, still working on getting a statistical inference method because we don't have good estimates for the rates. We've extended deriving the probability distribution from 4 taxa to 5. So now we have the probability distribution now for 5 taxon trees, which was quite a lot of work. And then from that, we've been able to we've got a start now on an SVD quintets algorithm instead of SVD quartets. As of right now, it's just proof by simulation. We can sort of simulate that this thing should be consistent, this SVD quintets algorithm. But unfortunately, there is just a ton of algebra to get to anything approaching a formal mathematical proof. So it looks like it's going to work, but we don't have a formal mathematical proof that it will be consistent and work. So that's the things that we're working on now as far as extending this. Shoot, went 2 minutes over time. Try to time this for 140, but hopefully, even moving fast, you learn something, and now hopefully you can ask a lot of questions about the method, about what we're working on now, or anything like that. So thanks for your time, and hope to answer some questions.